Hi all, welcome to Physio Exam Buddy. HCL secretion and its regulation is one of the most repeated physiology essay sub-questions. Let's revise the key concepts in just few minutes. HCl, it is basically secreted by the parietal cells or oxyntic cells which is present in the gastric mucosa. And in the purest specimen of the parietal cells, it contains about 0.17 normality HCl and a pH of 0.87. And regarding the concentration of the ions, it contains 100 milliequivalent of chloride and 0.00004 milliequivalent of H plus. And it is the H plus K plus ATPase pump which is present in the apical membrane of the parietal cells which pumps the H plus against a concentration gradient. And this is the structure of the parietal cells. Usually the parietal cells are polarized and it has got an apical membrane facing the lumen of the gastric glands, as well as there is a basolateral membrane which is in contact with the interstitial fluid, and there are certain canaliculi which extend from this apical surface into the cell. And this is the diagram you have to draw when HCl secretion is asked. And this is the this picture is the rusting state of a cell. You can see here multiple tubulovesicular structures, and on it you can see a blue structure which is our H plus K plus ATPase molecules in the walls of these tubulovesicular structures. And once it is stimulated, this parietal cells start secreting the HCl, and during that time these tubulovesicular structures, it will move to the apical membrane and fuses with the apical membrane of the parietal cells, and it will insert the H plus K plus ATPase molecules into the apical membrane. Once these ATPase molecules are exposed to these potassium ions which is present in the extracellular fluid, H plus K plus exchange begins. That is, K plus will be taken into the parietal cells, whereas H plus will be expelled out of the parietal cell. And you can also see here that chloride is also extruded down its electrochemical gradient through certain channels which is activated by cyclic AMP. H plus, which is extruded into the gastric lumen, it comes from H2CO3, and this H2CO3, it is formed by the hydration of carbon dioxide in the presence of the enzyme carbonic anhydrase which is present in the parietal cell. And this HCO3, it is extruded by an antipode which is present in the basolateral membrane, and this exchange happens with the exchange of another anion. Usually chloride is an abundant anion in the interstitial fluid, and the chloride will be the anion usually exchanged for this bicarbonate, and this chloride will enter into the parietal cell. The H plus, which is secreted into the gastric lumen in exchange for this potassium by H plus K plus ATPase pump, and in the basolateral membrane, HCO3 is exchanged for the chloride in the interstitial fluid by an antipode. And there is also a sodium potassium ATPase in the basolateral membrane itself, which will keep the sodium concentration inside the parietal cell very low. And the potassium, it is also pumped out through the potassium channels which is present into the lumen for providing the K plus which is required for the H plus K plus ATPase pump for the exchange of H plus ions into the gastric lumen. That's about the secretion of the HCl. And one physiological basis question commonly asked from this area is, what is postprandial alkaline type? When a question or physiological base is asked as postprandial alkaline type, you have to draw the diagram of secretion of HCl. And what is this postprandial alkaline type is that after a heavy meal, large amount of H plus ions will be formed in the parietal cell, and it is secreted into the canalicular lumen. At the same time, you have seen in the secretion of HCl that at the same time HCO3, it will be pumped back into the blood in exchange of an anion which is the chloride. So more and more amount of HCO3 will be released into the blood after this heavy meal. So what happens to the pH of the systemic blood? It will actually increase because bicarbonate is the alkaline pH, and some alkali will also be excreted through the urine and it became alkaline. So this condition, it is known as the postprandial alkaline tide, and please remember to draw the diagram of secretion of HCl in the postprandial alkaline tide. Now about the factors regulating HCl secretion. First of all, we will see the stimulants and next the inhibitors of HCl secretion, and this is the diagram you have to draw when the regulation of HCl secretion is asked. The most common stimulants include gastrin, which is secreted from the gastric mucosa, then acetylcholine from the parasympathetic nerves, then histamine from the endochromaffin-like cell. And out of these three, the gastrin, it is one of the most potent stimulus for the HCl secretion. First, about the gastrin. Gastrin is usually produced by the G cells in response to the products of protein digestion. And this gastrin will reach our parietal cell by an endocrine mechanism. And there are two mechanisms by which the gastrin increases HCl secretion. And the principal way is via the endochromaffin-like cells, which will stimulate the secretion of histamine and thereby again increasing the secretion of HCl. And another mechanism by which the gastrin increases HCl secretion is by acting directly on the gastrin receptors, and it causes an increase in the intracellular calcium level, which will activate the protein kinases and increase activity of H plus K plus ATPase pump in the apical membrane of the parietal cell. Stimulant for the HCl secretion is the acetylcholine. It is released mainly in response to the parasympathetic stimulation. Once the acetylcholine is secreted, it will combine or bind with the M3 muscarinic receptors, which is present on the parietal cell. It causes an increase in the intracellular calcium level, and which will in turn increase the protein kinases activity inside the parietal cell, thereby increasing the H plus K plus ATPase activity, and then the increase causes an increase in the HCl secretion. Next stimulant for the HCl secretion is the histamine, which is produced by the endochromaffin-like cells when in response to the gastrin binding to the endochromaffin-like cells. And this will act via a paracrine fraction and bind to the H2 receptors present on the parietal cells. And via a stimulated G protein, it will actually increase the activity of the adenyl cyclase enzyme. And there will be increase in the conversion of the ATP into cyclic AMP. So more and more cyclic AMP will be formed inside the parietal cell, which will again in turn cause an increase in the protein kinase activity, and then the H plus K plus ATPase activity, and finally increase in the HCl secretion. Next is about the inhibitors of the acid secretion. First one is the prostaglandin E2. Instead of a stimulatory G protein, this PGE2, it will come and bind with the receptor which is present on the parietal cell itself, but it will cause the activation of an inhibitory G protein. So this will be inhibited and produces a decrease in the HCl secretion. Next, the other inhibitors of the acid secretion include somatostatin, secreted from the D cells in the antrum, secretin, secreted from the S cells in the duodenum, gastric inhibitory peptide or GIP, then bombicin, H2 receptor antagonists like cimetidine, ranitidine, H plus K plus ATPase inhibitors, that is the omeprazole, and anticholinergic drugs, that is the atropine, usually inhibits the acid secretion. Other factors which decreases the HCl secretion are high H plus in the duodenum and as well as in the pyloric antrum, presence of fat in the duodenum, hyperosmolarity in the duodenum, and certain emotional factors like fear and depression is also found to decrease the HCl secretion. Finally, for completion's sake, what are the functions of the HCl? The main function is conversion of pepsinogen to pepsin. It will provide an optimum pH for the action of the pepsin. Bactericidal action is also there, which will kill the bacteria. Then stimulate the secretion of certain hormones like secretin as well as cholecystokinin. Neutralizes the alkaline secretion. It has also a role in the prevention of gastric carcinoma and also helps in the absorption of the iron. So that's about the HCl secretion as well as regulation. Thank you.